and last presentation for our U.S. Poultry funded research tech talks. I would like to introduce Dr. Brian Jordan, Associate Professor at the University of Georgia within the Department of Population Health. The title of Dr. Jordan's presentation is Investigation into the Cause and Prevention of False Layer Syndrome.
Thank you, Denise. Thank you all for being here today, the last talk of the day to kind of finish out this session of Tech Talks. And as Dr. Hurd said, we are going to talk about the cause and prevention of false layer syndrome in the U.S., which, as you may tell by the title slide here, is caused by a DMV 1639 type infectious bronchitis virus. All right, so false layer syndrome and cystic oviduct, those terms are kind of used interchangeably here in the U.S., is a documented but really not frequent presentation, mostly in table egg laying chickens, where the birds appear outwardly perfectly normal, right? They don't look like they're sick, they're fully fleshed, feathering looks very good, but those birds never lay an egg. They never come into production. And then when you open those birds and you look at the video that's not playing, there we go. When you open up that bird, you see that the oviduct has just become one giant fluid-filled cyst. Right? So the ovary actually is still fully functional. You still have the hierarchy of follicles. Oftentimes these birds are still ovulating. The ova just get absorbed into the body cavity, and so you'll have a very bright fat pad where that yolk is being resorbed, but that oviduct is incapable of actually capturing that ovulated ova there and forming a mature egg. All right, and there are a couple different things that can cause cystic oviduct formation, but the most common, if you go back through the literature, particularly in other parts of the world, is infectious bronchitis virus. Now, infectious bronchitis is a coronavirus. It's sort of a cousin to COVID. It's the COVID of chickens, I like to say. And it typically causes a respiratory disease, as we will show on the next slide, right? But it can affect other internal organs, just not as frequently, which is why we don't see this false layer syndrome as often as we do see the respiratory disease caused by bronchitis. There are some strains listed here that have been identified in the past to cause false layer syndrome. Massachusetts might strike a bell. It's the most probably known strain worldwide. It was the original bronchitis that was identified in the 1930s. It's used as a vaccine everywhere in the world, right? And it was sort of the original, so anything that could have happened, Massachusetts showed us the way. But then more recently, this Australian T strain, which is specific to Australia. Then you've got this QX strain, which originated in Asia and has actually progressed through many other parts of the world, not in North America though. And that QX strain is more virulent than other bronchitis strains that we typically have. So that one is a little bit different as well. But as I mentioned, the main influence of bronchitis virus as a coronavirus is this respiratory disease, right? So it infects the respiratory tract, the trachea, it causes cholestasis in the trachea. Cholestasis in the trachea then makes it so that that mucus there in the trachea can trap pathogens, but it can't get rid of the pathogens. And so those aerosolized bacteria, things like the bird is breathing in, are gonna make their way down the respiratory system. As the bird's breathing in, they're gonna move down to the air sacs. They replicate in the air sacs, causing air saculitis, and this will get your bird condemned at processing, right? So the inspector is on the processing line, bird comes through on the evisceration side, they see this widespread bacterial infection, that meat can't go to the plant, right? That meat is now lost. So bronchitis, as I mentioned, can infect other tissues, mainly the kidney and the reproductive tract. So other tissues that have a ciliated epithelium at some point in the development of that tissue. That's kind of the common thread, right? And so kidney disease, it causes urate deposition in the kidneys, a lot of flushing, dirty, wet litter, right? And when it affects the reproductive tract, when the birds are already in production, you've got this classic sort of wrinkled shell. The shells may turn pale, they'll be a different color, maybe reduced case weights, but then you've also got this cystic oviduct formation. Now, the egg influences, the reduced case weights, the wrinkled shells, things like that, that's what we typically think about bronchitis in egg-laying birds. Again, I mentioned earlier, we don't see this cystic oviduct formation very often. Historically in the US, we haven't faced it much at all. And I'm gonna tell you why in just a second. So starting in 2017 is when this issue really began, and it actually happened in Canada, not here in the US. So in Canada, there's 22 flocks of commercial layers, all breeds that they were used in Canada. The birds were supposed to peak 24 to 32 weeks, but you look at that production, 40 to 90% production drop, right? So a significant drop in production, meaning those birds never laid an egg. Again, normal outward appearance, some additional mortality, then these false layer cystic odor. This is a picture from one of the necropsies done in Canada, right? Again, at that time, this was sort of new. We hadn't seen this in decades, right? Everybody starts panicking a little bit. So we go back and we start doing some surveillance. What's happening? What virus is this? Is it QX? Because again, QX is sort of the most recent strain internationally that has caused this type of disease. Let's make sure we don't have that. This is some viral surveillance from the Animal Health Laboratory from Canada. And you can see over time, you know, in the early time period when this was being collected, it was 491 type strains, 793B strains of bronchitis. But you see over time this yellow bar, DMV. This yellow bar began increasing and peaked around that 2017 time period in Canada. Well, DMV 1639 is the strain name. It was actually identified in the Delmarva Peninsula in 2011. That's where the 11 comes from, that's the year. But it didn't really cause any disease to speak of until 2014, 2015. At that time, it was in broilers just in the Delmarva Peninsula. It caused kidney disease. So those pictures I showed you earlier of the urate deposition, the flushing birds, the dirty litter, there was increased mortality. It's a big deal in that 2014 to 2015 window. If we look at where DMV falls in relation to other strains that we have circulating in the US and Canada, you can see it's kind of up here in this other grouping of Pennsylvania type viruses that just kind of linger in the Pennsylvania area where they have a lot of layers and breeders, and then over in Delmarva where they have a lot of broilers. So where there's a mixture of different bird types that are kind of grown together in that area. And then as we continue down the timeline of this DMV 1639, we said in 2014-15 we had these significant clinical cases in broilers. But 2015 into 2016, the clinical picture actually kind of quieted down a little bit. We didn't have as much disease. We thought, okay, maybe this was just kind of a one-time thing. We've now got it under control. Then in 2017, these false layer cases really hit Canada hard, all right? Moving on 2017 to 2018, the false layer cases in Canada decreased. We began to see them in the US now. Then moving forward in time out to 2018, 2019, we had some lingering false layer cases in the US. But then in 2019, it popped back up again in broilers. And in broilers, it became strictly respiratory. We didn't see the kidney involvement anymore. And after 2019, it jumped out of Delmarva off of that peninsula, and it spread across the rest of the United States, and it is everywhere chickens are produced in the US, except California, but I think if we went and tested real hard, we'd probably find it there too. All right, so this virus has gone through the US. It's caused major problems actually in the broiler and on the layer side, right? And this led to this industry situation. No causative agent confirmed. And I'll explain why that bronchitis as the causative agent wasn't confirmed until much later, all right? So bronchitis was speculated. We thought it was DMV, but we didn't know. We couldn't recreate that at the time. No control strategy. There was no DMV type vaccine. Right? Companies were trying a lot of different vaccination strategies to see what would work, but it was just trying to make it stop from a production standpoint, right? There wasn't any real science or data to back up any of the things that they were doing, which led to this US Poultry and Egg Grant, which is the Board Research Initiative, BRF 012, investigation into the cause and prevention of false layer syndrome. So each year, as you may be aware, the US Poultry and Egg Association puts out a special call for I would say projects that are addressing a very timely and relevant issue. And this year, false layer syndrome was one of those. We put together this grant proposal. We were very fortunate to receive this grant from US Poultry and Egg Foundation. And so here's what we went through. So we divided this project into two stages. First, we wanted to confirm that bronchitis was actually the causative agent of false layer syndrome. And in particular, this DMV 1639 strain that we suspected but didn't know for sure. So to do this, we got 7 groups of 120 SPF chicks. right? So to get female SPF chicks, obviously we had to hatch twice as many. We had to do the sexing so that we just had females. And we were using specific pathogen-free SPF chicks because we didn't want maternal antibodies in these studies initially. We didn't want that influencing factor to mess with whether we were able to recreate the disease. We divided these birds up into different groups, and we challenged, either had our negative control, we challenged with a Massachusetts type virus, or a DMV challenge virus, a field virus that we had isolated in our laboratory at 3 different time points, 3 days, 7 days, or 14 days of age, okay? The classic presentation, if you went back to the literature from the 50s and 60s, was that the infection had to occur very early, that there was an age-related resistance. And so that's why we focused on these young time points. That is also why it was very hard to actually show that in the field, this DMV strain was the causative agent. Remember, if you recall back to the clinical presentation, you didn't know those birds were sick until they were 24, 25, 26 weeks old and they didn't lay eggs. If we're thinking the infection happens in the first couple weeks, that virus is long gone, right? There's a big window between actually seeing the clinical picture and when the infection and the challenge occurred. So to go through this data collection, 7 days post-challenge, we collected swabs just to confirm that the challenge was successful, that we got all of our birds challenged. We did necropsies at a lot of different time points to try to understand the evolution of the cyst. When did it form? Can we determine what is causing that cyst? There is still some unknowns there on whether it's just the inflammatory response, whether it is actual damage from the virus replicating itself. So we were trying to analyze that as well. And then we took 10 oviducts per challenge group and submitted for histopathology so we could look at the cellular change during this process. Right, so if we look at our results post-challenge, I'm just showing you this to show that all of our challenge groups were, except for one, 100% positively challenged. Very good CT values indicating a very successful challenge, very good viral load in these birds. And we were able to recreate cystic oviducts in a percentage of these birds. Okay, and I'll show that on the next slide. But this is what a typical cyst would look like at the different ages. So this is from the M41 challenge on the left, the Massachusetts. This is from the DMV challenge on the right. Right, cysts are very similar. We could see them induced at our different necropsy time points as early as 1 month post-challenge. So the cysts actually started forming quite soon after that challenge. But again, outwardly, no change in appearance. If we look at overall numbers as percent of total, from a 3-day challenge, the DMV challenge virus caused 25%, basically 25% of the birds had cystic oviducts when we added up all of our necropsy time points. At the 7-day challenge, that was the peak for Massachusetts at 32%, right? But in both cases, you can see that by 14 days, naturally, without any intervention, the prevalence of cystic oviducts had declined. So this did kind of confirm to us that there was an age-related resistance to some of this disease manifestation, and so that this challenge must be happening very early on for these birds. Now, something else that's interesting from this table is you may wonder, why were 100% of the birds, why did they not develop cystic oviducts when I showed you that 100% of the birds got challenged? And that's an interesting question that I don't know the answer to, but it mimics what we were seeing in the field because all of those birds were challenged pretty uniformly, and on average, about 25% was the rate of cystic oviduct if you kind of average all the flocks together that were affected. So this actually mimics kind of what was being seen in the field at the time. Why it's not every one, I don't know for sure, and that's still a question that we're interested in. But one thing that we did do is this riboprobe analysis, right? So this was a bird that was challenged with a DMV at 3 days of age. We collected the oviduct at 10 days of age, so 7 days post-challenge, using an RNA probe that was specific for that DMV 1639 virus. We were able to show that it was, in fact, the DMV bronchitis replicating in the oviduct that was causing the damage that then ultimately led to that cystic oviduct. So we fulfilled Koch's postulate in 2 senses. We could challenge and recreate the clinical presentation of that cyst, but we also showed molecularly by identifying that specific viral strain in these oviducts that it was that virus that was causing that, right? So we were able to confirm that. All right, so to conclude experiment 1, and you can see this paper is published, we had DMV can be the causative agent. I didn't talk about Massachusetts much, but it did exactly the same thing. So Massachusetts can cause cystic oviduct development as well. There is an age relationship. The birds have to be younger. I didn't show it in this data set. We had an older group that we challenged as well. We waited several weeks to challenge, and those birds didn't develop cyst at all, so I didn't include it here. The window for detecting bronchitis is very small, right? So a lot of questions when this was going on, I would ask, do you want me to send you oviducts? No, don't send me an oviduct if you're trying to identify bronchitis. Send me a trachea or a cecal tonsil. That's your best bet. The time that it's replicating in that oviduct is very, very minimal. And like I said, it's 7 days post-infection. There's no way you're gonna know to go randomly collect a chick and try to take that oviduct within the window when you might actually find that bronchitis virus. We proved that it was there, but it's not a great sample for doing diagnostics. So this led to the second aim of the study. Now let's figure out if we can use a vaccination strategy to help prevent this. Now again, the industry, because of the losses they were facing, they had changed their vaccination protocol, right? They had to. Our hypothesis, based off of what the industry had done, was that if we moved the first bronchitis vaccination from 2 weeks of age to the hatchery, that vaccination would then prevent the cystic oviduct formation, all right? And because this is what the commercial poultry industry, both in Canada and the US, had done and still do to this day. They just moved that first vaccination up to the hatchery to try to prevent infection. So here's our experimental design again. We used 2 different vaccine types, the 2 most common types that we use here in the US, a Massachusetts type, a Georgia 08 type. We then did follow it up with additional boost vaccinations to try to mimic a commercial vaccination schedule. We had positive and negative controls, and we necropsied at different time points along the way, right? So the design was very similar to the first study. We just added a vaccination to it, still using maternal antibody negative SPF chicks at this point. Collected pretty much all the same data, don't need to go through this. All vaccinated flocks were positive for vaccine virus 7 days post-vaccination, so we confirmed that vaccination was successful. We went through our challenge, and we can see that in all the groups that got challenged, 97, 98, 100% of the birds were challenged. But one thing you might notice a little bit here is the viral load in the birds that were challenged was not quite as high as in the study where they didn't get vaccinated. So that was checkpoint number 1. Okay, maybe the vaccine is able to prevent or influence that infection cycle a little bit. That would make sense with what we see in the field. However, we did still see cyst in a lot of these birds. The presentation of the cyst changed. We still did have birds that would develop these full cysts, but we also had a lot of birds that would develop, it wasn't a fully formed bag-like cyst, but it also wasn't a normal oviduct. There was additional fluid in that oviduct, right? And if we look at totals on these between fluid and cyst, we see the overall percentage of affected oviducts is really the same between your vaccinated and your non-vaccinated groups. But what it did is it reduced the severity of that clinical presentation. So it went from a full cyst to just that oviduct that had fluid in it. From a research standpoint, this is pretty cool. Vaccination is doing something. From a production standpoint though, neither one of those birds are laying eggs, right? Neither one of those birds in these groups are laying eggs. So it didn't really matter from an actual clinical endpoint, they were still able to produce cysts, all right? And if we go through, just look at these. So the conclusions, day 1 vaccination influenced the severity of the cystic oviduct, but again, and the vaccine serotype was not important. Didn't matter if it was Massachusetts, if it was Georgia 08, either one, both of them worked about the same. Interestingly, in one of our groups that got vaccinated and did not get challenged, and we confirmed that they were not exposed to that DMV challenge virus, they still developed cystic oviducts, which tells me that in some cases, the vaccine by itself can induce that same cyst. These are live vaccines. It could replicate in the oviduct. It was a small percentage, but it did happen. Day 14 vaccination did not influence the development of cystic oviduct. So we had one group that didn't receive a day 1 vaccine. They just got a day 14 vaccine. That group was no different than our non-vaccinated group. Again, confirming what happened in the industry during the time of the outbreak. But this was different than what we saw in the field. for the commercial companies, once they moved that vaccine from 2 weeks to day 1 in the hatchery, their prevalence of cystic oviduct basically stopped. I mean, it was kind of night and day. It stopped, all right? We were able to show that DMV was the challenge virus, but our presence of cystic oviduct didn't stop, right? We still saw cyst in those challenged birds. So what is the real difference in between what we did and what the industry situation was? Question for the audience. What was the difference? Maternal antibodies, right? We did our work in SPF, maternal antibody negative birds. Commercial pullets are maternal antibody positive. Now, the standing sort of the school of thought for bronchitis and maternal antibodies is that maternal antibodies don't really influence infectious bronchitis virus infections, especially not vaccinations, because we vaccinate every boiler produced in the world basically with IBV at day of hatch, right? And that is true. But what kind of antibodies are maternal antibodies? Does anybody know? IgY, same as IgG, circulating in the blood. What is the antibody that actually prevents bronchitis infection? It's IgA. And IgA is produced in the Harderian gland behind the eye, right? But what IgY can do is prevent viremia. So the birds still get infected, but that IgY will prevent the virus from moving around the body to other organs and other tissues. And so this actually led to a third experiment that wasn't part of the original project, but with the data we produced from the US Poultry and Egg Grant, we were able to partner with one of our commercial egg producers to look at the role of maternal antibodies in combination with vaccination in the development of false layer syndrome. And so our hypothesis here was that that maternal antibody, in addition to that vaccine, that you had to have both, would totally prevent development of false layer syndrome. May not prevent infection, but it would prevent the development of the syndrome. So we designed a study here, and we actually partnered with another lab out at UC Davis as well. And so we used one type of vaccine, the Massachusetts type. They used a Georgia 08 type of vaccine in their facility in this partner study. And we again included birds that were maternal antibody negative as well, just to make sure we could recreate the disease with our challenge. And just looking at the data, what we saw is, if you look at the red bar is the maternal antibody negative birds, gray bar is maternal antibody positive birds, right? So in our maternal antibody negative birds, red bar, that were vaccinated and challenged, when we added maternal antibodies to the gray, you can see that we had a 50% drop in that cystic oviduct presence. Same thing was true in birds that were not vaccinated. So the maternal antibodies alone, even without a vaccine, still did help. Didn't prevent it overall, the prevalence was higher, but it did help control the maternal antibodies. Now, interestingly in this case, right, the last study I told you that the vaccine by itself could cause some cystic oviduct. Maternal antibody negative birds. We have maternal antibody positive birds. That takes care of the vaccine situation. You're not going to get the viremia and introduction to other tissues. No cystic oviduct seen. So, prevention and control to finish up the false layer syndrome situations. Threefold strategy. You have to have maternal antibodies in those pullets. They don't have to be type specific. They don't necessarily have to be DMV specific, or if you're looking internationally QX specific or whatever strain it is. They just need to have maternal antibodies that can help bind up that virus so that you don't get the viremia and go to other tissues. Number two, you have to have that day one vaccination if you're having an outbreak. And it's not a cross protection situation, okay? You're not really achieving true cross protection. What you're doing is creating viral interference. You're giving a live vaccine at day one. That vaccine is infecting and replicating all the same tissues that your challenge virus would want to. So it basically just pushes the challenge virus out to a point where either the infection is very low, or you get into that window where you've got that age-related component past 2 weeks where the birds are just kind of naturally, able to overcome that. And then three, limit early exposure to challenge. You think about a challenge at 3 to 7 days of age. I mean, that's basically at chick placement. That's a very, very early challenge. So we need to tighten our biosecurity protocols, think about what we're moving in and out of those pullet houses in those first 2 weeks. We take care of that, and the rest takes care of itself. That's all I have. Thank you very much. Special thanks to Adria Mueller, who was my PhD student who did all the work on this project. She's now a DVM student at the University of Georgia, so she's a dual DVM PhD. Great student. She did the lion's share of all this work, so I have to give her credit. Any questions?
Dr. Jordan, I have a question for you as well.
Yes,
so
ma'am.
just out of curiosity, what are your theories on a quarter of the infected flocks getting cystic ovaries that haven't been vaccinated? You said that's something that you still haven't identified, but you're interested in looking into. So,
Right. So it's an interesting question because even when we do challenge studies in broilers, let's say, with bronchitis virus, not all the birds respond in the same way. Some birds have much greater clinical signs than others. Which is why, and if we think about cystic oviduct, it is a clinical sign or lesion if you want. It's just greatly delayed, right? When I didn't show the data here, but if you go through and you look at the actual viral loads in each of those birds, because we swabbed them too to measure infection, the viral loads are no different in a bird that has cystic oviduct or not. So it's something within the bird and their different response. And then I know there's a lot of work going on now looking at like MHCs and different immune factors and maybe even the genetics plays a role into it. I don't know, but we see it across layers, breeders, and broilers in that every bird doesn't uniformly respond to a challenge in the same way. And so it's something in there.
thank you. Any other questions for Dr. Jordan?
Yes, sir.
So does the virus only replicate in epithelial cells?
Yes, epithelial cells.
Thank you.
Yep, that's the target tissue for bronchitis. Yes.
Additional questions?
What was the breed of the layers used in your previous study?
So the SPF birds were hyaline, and the maternal antibody positive pullets were aviagen.
All right. Any other questions? All right. If not, thank you very much, Dr. Jordan.
Thank you. Thank you all for being here.